Hello, I am Alexey Kazakov. Today I present ENIGMA Genome Browser, one of ENIGMA data resources. You can access Genome Browser on the iseek.lbl.gov server. This server has about 10 genome collections powered by Genome Depot, a software platform for genome management, which was developed in ENIGMA. And the ENIGMA Genome Browser is two of these collections. One is a collection of NCBI genomes, including all published ENIGMA genomes. And the second is a collection of published and unpublished ENIGMA genomes and some other genomes for comparison. So to access this collection of unpublished genomes, you need a login and password that you can get from ENIGMA intranet site. So today I will use public genome collection as an example. Clicking on a link on the index page, you'll go to the start page of the genome collection. It has 8 buttons and a menu in the top left corner for navigation on the site. The menu is accessible from each page of the genome browser. Click on Genomes button, opens a list of genomes and the taxonomy profile of the genome collection and the search field, so we can search by name, or you can choose from the list. Taxonomy profile of the genome collection is interactive, so I can go up and down by taxonomy by clicking on one of the rings, or return to the root by clicking on the central circle. Or I can click on one of the names and it will open a new page for this taxon where I can limit the number of genomes by only genomes for this taxon by clicking on the link at the bottom of the page. For example, I need 3H11 genomes. I just type 3H11 in the search field of the genome page and with this. The top part of the genome page shows general information. Then there is an interactive genome viewer, search fields, and at the bottom, a section of data export links. Interactive genome viewer lets me scroll genome upstream, downstream, switch between scaffolds, zoom in to see nucleotide sequence, and view details for each gene or element in the genome. There are separate tracks for coding genes, operands, pseudogenes, and RNAs in the genome viewer. Search fields in the genome page let me find genes by one of functional annotations, or by locus tag, or by gene name, or by config name, or I can use one of functional classifications. For example, I can search by name of KEGG ortholog group, or if I don't know exact KEGG ortholog ID, I can open a list of all KEGG ortholog group mappings of this genome. When I find a group of interest, I click on this name, and it will show me all genes in this genome mapped to this ortholog group. The same way I can search in KEGG reactions. I can search in enzyme names or EC numbers, in transporter families, CASI families, or COG classes. I can also look at the list of Go terms mapped to this genome, or if I am interested in a specific KEGG pathway, I can open a list of KEGG pathways for this genome and find all genes which map to this pathway. For pathways, there is a link for visualization of KEGG maps in this genome. It opens a list of genes for the pathway and loads from the KEGG website a KEGG map. I can hover mouse pointer over the name of the step, find the identifier of KEGG ortholog group, and then find this group in the table and find the genes which are mapped to this step of the pathway. By clicking on the gene name, I go to the page for this gene. On this page, there is a general information about the gene, list of ortholog families mapped to this gene, an interactive genome viewer is this gene highlighted, a list of functional annotations. On the left, there are annotations generated by ENIGMA mapper, which are mappings to Go terms, KEGG reactions, KEGG pathways, and so on. And on the right, there are annotations generated by different annotation tools. For example, GapMind, HMM search with PFAM domain, or TigerFam. This is a list of ortholog family mappings for the current gene. These families have a different taxonomic scope. So the family with a narrow scope have a smaller number of representatives. This family has only 19 genes, only from Burkholder-Realis genomes. And on the family page, there is a functional profile that aggregates all functional mappings for representatives of this family. For KEGG reaction classification, KEGG pathways classification, from PFAM domains, from genome annotations, and others. This is interactive plot. I can zoom in on a classification of interest. It would show me a value, which is a number of genes with this function annotation. And at the bottom, there are links that save these annotations or protein sequences for this family in text files. For example, I can click to export functional profile as a tab-separated text. And then I can open this text file in a Google spreadsheet. The exported table shows me number of functional classifications, functional categories, textual descriptions, and number of genes in this family mapped to this or that functional class. Now I go back to the gene page. Next to the list of the names for ENIGMA families, there is a link for visualization of genome neighborhood based on these ortholog families. So these are representatives of the selected ortholog families in each genome, up to 50 genomes. I can say limit this number to 10 genomes, but show a wider area and click refresh. So this is a visualization of the region and a phylogenetic tree of the proteins from the selected ortholog family. I can export this image as PNG plot. I can export multiple alignment of protein sequences and the phylogenetic tree. And also it shows me a functional profile of all genes in this region. There are probably now too many of them. I can limit this region size to 5,000 base pairs. And it will show me that most of the genes are for amino acid transport and metabolism or single transduction in this region. Now I return to the gene page. I can export protein sequence of this gene by clicking on this protein link, or I can save the nucleotide sequence. If I click on the gene in the genome viewer, scroll down, there is a ORF sequence. Alternatively, I may want to export upstream region. Then I select this region in the interactive genome viewer. Click on the reference sequence dropdown and choose Save Track Data. Here I choose visible region option and click view. It shows me the nucleotide sequence of the upstream region of this gene. To find more homologs of this gene, I can select the protein sequence and copy, and then from the menu, go to protein sequence search, paste the sequence, and it will run BLAST-P to find all homologs in this genome collection. This is a list of hits. It shows gene ID, genome name, target genome taxonomy, function, identity percentage, and alignment length. At the bottom of the page, there is a link to export of the search results in tab-separated format and open in Google Spreadsheets. This text file contains a table of BLAST-P search with starts and ends of with number of mismatches and coordinates of the aligned region. Or I can copy the nucleotide sequence, go to the nucleotide sequence search page, and run megablast of the gene nucleotide sequence against the genome collection. This page also has a link to export full search results in tab-separated text format, or I can click on the hit and it will open a genome page with a highlighted region corresponding to the BLAST hit. The Genome Depot genome import pipeline generates operand predictions. So there are operand pages. An operand page contains a list of genes which are members of the operand. A genome viewer is the highlighted operand, and a link to comparative analysis tool. This tool finds orthologs of all members of the operand and displays a taxonomic profile for the genomes that contain orthologous operands, and also a functional profile of all orthologous operands. With the functional profile, it is possible to estimate conservation of gene functions in these conserved operands. E. coli genomes in the genome browser also contains regulatory information. There are predicted regulons based on profiles from RegulonDB database. Clicking on a link on the name of the regulon will open regulon page of one genome. It will show a list of sites and a list of genes which are regulon members in this genome. Also, a list of regulated operands. And there is a tool for a comparative analysis of regulons based on orthology of regulatory proteins and regulon members. I click on one of the links in the Build Conserved Regulons section. It would find the orthologs of this regulatory protein and check if this ortholog had regulon predictions. There are several E. coli genomes in the genome browser. It will display which of the regulatory sites are conserved across these related genomes. And also shows a list of identified conserved regulons and sites. In addition to search by genome name, it's also possible to search by strain name or strain metadata or ENIGMA isolates. On the strain table, there is a search field. For example, I can enter an isolation condition to the search field on the strains list, say aerobic, and find a list of strains that have this word aerobic in the metadata description. These data are imported from ENIGMA isolate browser or from spreadsheet exported from Coral. And the text search page lets me find genes across all genomes. I can search by gene name or product. I can search in gene annotations across all genomes, or I can find all representatives of this or that KEAC ortholog groups or the pathway. On the search page, also there is a field for search in taxonomy, or I can search for eggnog by eggnog family ID. For example, I want to find all genes from NOS-Z clade 2 family. This search took about 90 seconds. The resultant table shows me the list of genes that has NOS-Z2 annotated by PAMA. There are 73 genes in total, and I can export these proteins in FASTA format, or I can export these genes as tab separated text. So I choose text, and then I open the text file in Google Spreadsheet. It shows me locus tag, name of gene, name of organism, identifier of the genome, contig, ID, gene coordinates, strand, functional notation, and PAMA annotations. And finally, if you need additional information about the genome browser, on the third page, there is a documentation link that opens the online documentation website with a user guide that has a complete description of all pages and functions in the genome browser. Thank you very much.